For over a century, scientists have been learning about the arrangement and behavior of the central nervous system. This knowledge has led to discoveries allowing clinicians to modulate brain function through electrical stimulation. This paradigm of electrical stimulation is at the core of a novel set of therapeutics for mental disorders such as Parkinson's disease, essential tremor, epilepsy, and more recently, in chronic neuralgia. In a nutshell, stimulation involves sending bursts of small currents to the brain over a period of time on the order of months. The brain can be stimulated with implantable devices called microelectrode arrays, or MEAs for short. One pitfall of the MEA strategy is the brain's natural immune response to a foreign object, which is called the foreign body response. Scientists have been reducing the shape and size while increasing the flexibility of MEA devices so the foreign body response is not activated as quickly. The use of a material known as amorphous silicon carbide, also called ASIC, allows the fabrication of much smaller and more pliable devices. These smaller devices constitute a subset called ultra-microelectrode arrays. Normally, when we want to quantify the foreign body response, we take an image after staining cells with immunohistochemical biomarkers such as GFAP at different implant depths, and then measure the intensity of the immune response. With new UMEAs that cause very small foreign body responses, it is very difficult to identify the implant site and measure these small effects. This is the problem we addressed with this study.
So in this study, we did a semi-quantitative topological analysis, like local intensity surface roughness parameters on brain slice images. With this new analytic technique, we compared the immune response between small ASIC UMEA devices and available standard large silicon-based MEA devices. So our objectives were to 1. Compare the foreign body response between small ASIC UMEAs and larger control probes, and number 2, to evaluate the magnitude of this foreign body response between UMEAs and control probes across different cortical depths by using surface roughness parameters.
To briefly discuss the methods, we implanted adult rats with either ASIC ultramicroelectrode arrays that were fabricated at UTD or a commercially available silicon-based MEA. After 16 weeks, we perfused the animals with paraformaldehyde, then extracted, sliced, and stained the brains. Wide field images of brain slices were obtained with a confocal microscope. More details about the procedures can be found in the Materials and Methods section. We wrote scripts to automate the process of analyzing images in ImageJ.
So the plugin Surface J in ImageJ characterizes the surface roughness of the specified region of interest for the confocal images obtained from the methods, and the parameters that were obtained from Surface J are shown in Figure 4, and the respective mathematical equations from which they are derived are located in the appendix. To discuss the most significant results, figures were split into 3 different depths: superficial, middle, and deep. This is based on previous studies such as Citation 6. Looking at figures 4a, b, and c, the parameters RC, RA, and RQ were higher in the control probe compared to the ASIC UMEA probes across the superficial, middle, and deep depths. On the other hand, the parameters RSK and RKU were higher in the UMEA probe compared to the control probe across all 3 depths. One surface roughness parameter of particular interest is the mean height, or the RC value. RC is a measure of the mean height of pixel intensities. Therefore, images with a high magnitude of pixel intensity causes a greater RC, and by extension, a larger magnitude of immune activity. As seen in Figure 4a, we found that the mean height, or RC, derived from implant tissues was significantly higher for the larger surface area control probes, as compared to the UMEA ASIC probes with the smaller surface area. This essentially indicates that the larger surface area produces a higher foreign body response. In addition, we also observed a depth-based variation in the foreign body response across both probe types, which indicates a differential expression of GFAP with implantation depth. This observation was also reported in another study cited in the text. Overall, this data suggests that the small ASIC UMEA devices evoke a smaller immune response compared to the larger control devices. In regards to future studies, there are a number of different ways to describe the foreign body response, which can improve our understanding of how ASIC devices behave in vivo. These include macrophage infiltration, in regards to diapedesis or leukocyte extravasation, blood-brain barrier damage, and other second-order neuroinflammatory reactions which affect neural implants' ability to chronically stimulate cells. Thank you for listening.